Punnett squares are used in genetics to determine what the possible offspring could be from two individuals. Today, we're going to use a simple dominant recessive relationship, and we're going to look at fur color. For this simple dominant recessive relationship, we'll have a dominant allele indicated by a big B and a recessive allele indicated by a little b. Our big B will code for black fur, and our little b will code for brown fur. So, what I'd like to do is take a heterozygous individual and cross that with another heterozygous individual. I'm going to use two different colors so that you can follow the alleles. So the next thing we're going to do is draw our Punnett square. Which is just 4 boxes. We'll put the alleles for our first individual on top, big B, little b, and we'll put our alleles for our second individual down the side, big B, little b. How you work a Punnett square is you look at the box and you put the allele that's on top in the box and the allele that's on side in the box. So this one, we're going to drop down our big B and we're going to bring across our big B. So the genotype for this individual would be homozygous dominant and the fur color would be black. For this individual, we have a big B and a little b. For notation's sake, you always write the capital letter first. So we would write this big B, little b. In this box, we have big B, little b. So again, our capital letter goes first. And both of these individuals are heterozygous. They have one dominant allele and one recessive allele. But their phenotype for all three of these individuals is still going to be black. For this box down here, we have a little b and a little b. So the genotype for this individual is homozygous recessive, and the phenotype is fur color brown. Another part of this equation is we always look at probabilities and we kind of try to put this in numbers. So we could say that 3/4 of our offspring are going to have black fur and 1/4 of our offspring is going to have brown fur. Another way of writing this would be in a ratio. So our offspring would show up in a ratio of 3 black for every 1 brown individual. We can also do what's called a test cross. And for a test cross, you take an unknown individual. Sorry about that. For a test cross, you take an unknown individual and you try to figure out what their genotype is. So for this test cross, I'm going to take a black individual, a black fur, and I'm going to cross it with a brown fur. Here's the problem. For black fur, we can either have the genotype of homozygous dominant or heterozygous. For brown fur, there's only one genotype, and that's homozygous recessive. So when I have a test cross, I'm not going to know one of the genotypes. So how I would write this is B with a question mark and brown, which is homozygous recessive. When I go to do this cross, what I would do is I would look at which offspring I got. If I had a big B, and I mixed it with a brown individual, okay, so I have a homozygous, if my unknown is homozygous dominant, all of my offspring are going to end up heterozygous, and all of my offspring would have black fur. If instead of homozygous dominant, my black individual had a genotype of heterozygous, these offspring would end up being heterozygous over here, and they would have black, but these offspring are going to end up with brown fur. So I would have half black, half brown. And that's called the test cross. You match an unknown genotype that's dominant with the known genotype, which would be recessive, to figure out what your unknown genotype is. We'll be doing some practice with these in the lab. We'll just be doing them figuratively. We're not actually going to be breeding organisms in here, but it's a simple way to test what your unknown genotype is.